Thank you for the introduction, and also I would also like to thank the organizing committee for choosing our work here to be presented here. So I will talk about some recent efforts we've been doing to understand variation in single-cell transcriptome data sets, and I want to start with a very brief overview of what single-cell transcriptomic actually is. I think in this audience we can be brief here. So really the key idea is that previously in normal RNA sequencing technologies we've been assaying a large number of cells in bulk. So typically these are hundreds of thousands of cells or more, and hence we are only looking at average changes between samples or individuals that we're going to be able to study. Now using recent technologies, for example, such as this fluid-amp system for Illumina, but really the technology I talked about and the methods will be applying to all the single-cell technologies that are now being available. We can assay the transcriptome of single cells at the resolution of individual cells rather than bulk assays. Now, on the one hand, that is a really exciting opportunity. We can ask new questions that previously were hidden. We can look at questions like differentiations of cells. And these are things that really ultimately happen at the single cell level, so they're hidden if you look at bulk data sets. We can also ask the questions about what are cell types and subcell types that previously were hidden. But there are also new challenges, and one of these challenges that vary for many reasons, and there's different sources of heterogeneity. For example, they cycle, they go into apoptosis, they die, they might be stressed, and hence we need new tools to try to dissect these sources of variation and make sense of the data sets we're obtaining. I have here a cartoon that illustrates a problem that we're trying to solve. So here at the top, you see a differentiation trajectory. That's what we really want to do. So here in this cartoon, every dot represents a cell. We actually ultimately want to order cells along the trajectory from naive cell types to differentiate cell type. This is here Th2, and that's the data set that I will talk about later on. So we'd like to order these cells by the expression of a marker gene, so GATA3 that I'm showing here, and that's a typical differentiation marker. GATA3 low corresponds to naive T cells, and GATA3 high to differentiated T cells. But the challenge is we don't really see these types of data that I showed here, but rather these cells undergo different processes and heterogeneous processes at the same time, and one of them is that they cycle. For example, some of these cells are in G1 cycle state, others in G2M and in S, and hence the actual expression profile that we're measuring in practice looks more something like this, what here's at the bottom. And as you can anticipate, if we were to order the cells based on the GATA3 expression profile here at the bottom, we would get to very, very different answers than the clean data set that we've shown you at the top. Now, could you argue this is a rare example, but we actually found that this is not the case. So here on the circus plot showing correlations between cell cycle genes. These are genes that are annotated with the cell cycle, so they track cell cycle stat and the whole transcriptome, and you can see that there's widespread correlation. So many genes will look like this GATA3 example, and hence if we do transcriptome analysis without removing that source of variation that we maybe don't care about, which in this case is the cell cycle, we would come to very, very different conclusions. We'd come to different differentiation trajectories, and our inferences about the data sets will be quite different. So the good news is this problem, this challenge of heterogeneity and removing variation we don't care about to understand what we do care about is not new. So actually in the past we've been looking at methods in genetics where we might be interested in the variation of a particular variant in the genome. Here at the top, and we try to understand how does this variant affect the expression of a particular gene. And actually also in this application, where we don't look at variation between cells, but between individuals, there has been the challenge of removing variation, things like population structure, batch effects, unwanted variations. And really what our work is about is to leverage these tools from genetics, where we want to understand variation between people, to single-cell genomics, where we try to understand variation between cells. So how do we do this? So what we do in practice is we actually try to estimate a cell-to-cell covariance matrix. This is this matrix here at the top. That's a matrix that cell by cell captures the similarity between cells due to the process we don't care about, in this case cell cycle. Once we've done the inference, then we can try to account for this variation and solve interesting questions. We can ask what are the sources of variation, so dissecting single-cell transcriptome variation. We can also solve problems like gene correlation network analysis, and we can look at questions like are there new subcell types that previously were hidden if you don't account for the cell cycle effect. So the key trick that we're using is that we have good markers of cell cycle, and I already mentioned this earlier on. So we know genes that are tracking cell cycle status, and that's showing up here in the top right. So we're going to use the set of genes and transcripts that track cell cycle status and only model this slice of the transcriptome matrix. So the assumption we're making is that the set of genes that are annotated to cell cycle processes, that these are independent, conditioned on a suitable covariance matrix. So this was this formula he expresses. So this slice of the matrix is independent across genes. All these genes come from one big model, and that model is a random effect model that is effectively governed by this cell-to-cell covariance matrix as shown here at the bottom right. So an intuitive way to think about this model, it's something like doing PCA on this slice of cell cycle genes I'm showing here at the top. I'm sorry. And effectively, the cell-to-cell covariance matrix is governed by the first principal component. So it's a low-rank model that we're fitting to the data. The other important thing to notice is that we don't need to know the cell cycle status. The only thing we need to know is which cells are in the same state or in different states. So the similarity encodes just similarity in status rather than exact assignment, which is an important component. There's one quick thing we need to solve up front, which is technical noise. I'm not going into much in detail here, but what we do, we leverage technical spiking. This is work that's been really pioneered by Simon Anders and the Brendig et al. Paper 2013 in John Marioni's lab. And what we're doing is effectively we're spiking in exact same quantities, spike-ins here shown in blue, that shouldn't vary across cells, but they do in practice, and this variation is simply due to technical reasons. We can then use this technical variation to actually estimate for all our other transcripts what's the background noise, what is the variation due to technical reasons, and we can propagate that information in our models to get better inferences about the actual process we're interested in. So we propagate this technical source of noise through our models. So what we can then do once we have the cell-to-cell transcriptome covariance matrix, we can ask questions about what is the source of variation, why do genes vary in the transcriptome. So effectively we can fit an ANOVA model where we try to dissect the variation due to the cell cycle covariance, which we now just learned from the data itself, due to residual biological processes we might be interested in, and technical noise, which we get from the spiking I just mentioned earlier on. Before proceeding, the first question for us was, does it actually work? Do we really capture cell cycle information using this relatively simple approach? We were fortunate that in Sarah Teich's lab, Kedar has been producing a staged ES cell data sets of 300 cells that is experimentally annotated for the cell cycle. So using hex staining, we know which cells are in G1S and G2M. What I'm showing here is just a PCA plot of these 300 cells. And what we then set out to do is to say we apply our method without giving it information about the cell cycle state and compare the results when we use the experimental ground truth. And this plot here shows you the explained variance on the x-axis for every single gene, so every dot is a gene that we can attribute to the cell cycle using our model approach, doesn't know about the cell cycle status. And the y-axis, the explained variance using the actual experimental ground truth, and you can see these are quite nicely correlated, suggesting that really our model explains cell cycle and not other sources of variation between cells. We can also use the model, and that will be very important for the subsequent analysis I'm showing, to remove the cell cycle effect. So here on the right, you see the same PCA analysis and the data on the left. After removing and counting for the cell cycle effect for our model, and as you can see, the signature is almost all gone, except for maybe some variation in the S phase, which actually turns out to be the most difficult cell cycle state to cover, but this is a much reduced effect due to cell cycle. We next set out to look at actual questions, and this is Th2 differentiation that I tried to model in the beginning of my talk. So we looked at data sets that have been generated by a combination of cells on this lineage here, differentiating from naive T cells to Th2 cells. We have 92.6 cells, profiled using the C1 system, and we were expecting to find populations that are either in the naive state or the Th2 states and some combination between these two. When looking at the explained variance using a model, so I'm binning here genes by the proportion of variance explained by technical noise, which I mentioned earlier is a big challenge. These genes here have 10% technical noise, and here on the right hand we have basically no technical noise. We can see that overall the cell cycle here shown in orange explains the largest proportion of variance after technical sources of variation. It's also interesting to see that because of this cell cycle variation, we see a reduced variation actually in concordance to bulk. So what I'm showing here is the ranking of genes on the x-axis by variation between cells. On the y-axis, the enrichment of genes differentially expressed between these two populations, naive and Th2 cells. We can see if we regress out the cell cycle, there is clear concordance that variable genes are differentially expressed between the population. If you don't regress cell cycle in green, there's no concordance, again showing, suggesting that cell cycle really masks interesting processes that happen at the single cell level, and we need to account for the process to understand the biology. Now, doing this, we can now go ahead and ask questions about our single-cell transcriptome profiles. The first thing we looked at are gene-gene correlation networks. So we're correlating here genes with genes on this big matrix. And if we do this on the naive data, we find very large-scale correlation networks. 500,000 genes are correlated in some sense. If we count for the cell cycle, these networks are much, much more sparse and concentrated on 20,000 edges. And the processes that we find are much more enriched for T-cell biology. So that actually suggests that us to be doing something useful. Perhaps the most interesting question, however, is do we find new subpopulations? So what I'm showing here is a PCA analysis of these 96 cells, again on the raw data on the left. If we now account for the cell cycle, we do find two subpopulations. And I'm coloring here these subpopulations actually by GATA3. So it really turns out if we overlay this marker genes that is an indicator for T-cell differentiation, the left cluster is GATA3 low, i.e. these are naive cells, and the right cluster is GATA3 high. These are differentiated cells, and really suggest that by accounting for the cell cycle, we are able to recapitulate these two populations that we would expect intuitively. We not only found GATA3 differentially expressed, but a whole battery of T-cell differentiation markers. There are 401 genes that include many of these cell surface marker, IL-4A and others, that we expect to see in T-cell differentiation processes. I want to give an outlook of where these models can be used in addition. So we can not only model cell cycle, as done here, but we can also go back and ask, can we look at the differentiation signature itself that we now identified, these 401 genes that are differentially expressed between these two types of populations? And the answer is yes. We can apply the same principle, use a second gene set, now one that accounts for differentiation processes. And then we can dissect the variance in our transcriptome data and looking at these two components at the same time. So ask the question in an ANOVA model, how much variance does cell cycle explain? How much variance does the differentiation process explain? And perhaps even, is there an interplay between these two processes that are happening at the single cell level between these two entities? And that's what I'm showing here. So this is the same type of variance decomposition. Again, I'm binning genes here. The top bar actually shows the density. And you can see that differentiation in blue explains substantially less variation than the cell cycle in orange. And that also explains why you had to take account for the cell cycle contribution in order to actually see the differentiation signatures if you do something like PCA. Now, as mentioned, we can look at interactions. That's what I'm doing here in blue. So this is a linear interaction model between these two processes. These are genes where the cell cycle has an effect on the transcriptome in a differentiation-specific manner where these processes are interlinked, which makes sense. T-cell differentiation is a proliferative differentiation protocol, so these cells have to undergo a few cycles before they start differentiating. And these genes are enriched for positive cell population and negative regulation of apoptosis. I want to give you one example of what that actually is an interaction. So this is BATF. This is one actually a differentiation mark in Th2. And what you see in the y-axis here is the expression of this gene. The x-axis shows our inferred cell cycle factor. So this is G1, this is G2M, this is this continuous scale that our model teases apart. In color, you see the differentiation status, where again, red corresponds to differentiated cells, blue to undifferentiated cells. But again, none of these two data sets are available. So we neither know the cell cycle status nor the differentiation status. That's all model inferences. And what I'm showing here is the regression line between cell cycling expression, where the red population, the differentiated cells, have a much steeper relationships, suggesting that cell cycle is more important for expression variation, versus the naive cells in blue are less interlinked with the cell cycle. That gives you an idea of the type of interactions and processes we can tease apart using model-based inferences, such as this model we're proposing. Now we can generalize this to more processes and other domains as the data set's increasing in single-cell genomics. This is 96 cells. Now with DropSeq, we can look at hundreds of cells and thousands of cells, and that is now an area going forward. I also want to make one quick link to the future, where I think these technologies will be really important, and that is when we'll be able to assay multiple omics layers from the same cells. This is unpublished data from Wolf Exlag in collaboration with Thierry Wood, where we have now a physical separation protocol where we can take a single cell and separate DNA and RNA from the same cell. And that enables looking at transcriptomes and epigenomes from the same identical cells. I'm showing here at the bottom is a gene ESRB, where we can look at heterogeneity on the methylation side here on the x-axis. So every dot here is a single cell. You find regions where cells are unmethylated, highly methylated. And we can now look at correlations between the epigenome and the transcriptome, where we have interesting about linking epigenetic transcriptome heterogeneity. Here in this case, we find two regions, ESRB, expression methylation variation, P300 region, and actually, even that we only have 22 cells at present, we find quite a few of these types of correlations genome-wide where the insync process is interlinking. And with this, I want to summarize. The key take-homes are that there's a small number of genes with annotated to known processes like a cell cycle or differentiation processes are actually sufficient in many cases to recapitulate cell-to-cell covariance matrices. These covariance matrices can be used to understand the source of variation. They can be used to look at interactions between processes, and they can be used to remove sources of variation you might not be interested in as a cell cycle if you, for example, want to study T-cell differentiation. Now, if you account for the cell cycle covariance, we can actually uncover new biological processes. One of these applications is more compact gene-gene correlations. We can look at cell subclusters, and we can detect interactions between genes. And this would be very interesting if we get other omics layers, such as methylation, that I showed you as an example. I want to finish with a quick note that actually that shows also the highlight of how different fields interact. Really, these methods are one-to-one application of tools that have been developed in statistical genetics. Variation between people is the question of interest. And here we really translationally applied this, and this maybe gives an example how different fields could interact also in this venue, like at ISMB. With this, I want to acknowledge people who have done the work. So really, Florian Büttner has been driving this. He was first at Helmholtz and moved to the EBI. This is a close collaboration with John Marioni and other colleagues at the EBI in Sanger, Wolf Reich, and Gavin Kelsey and Thierry Wirth for generating this epigenome data I briefly mentioned here. You can find the manuscript over there. There's software and tools. This SELVM, our approach, is here available on GitHub. And with this, I thank you and look forward to questions.
Thank you very much. So, any questions?
Your Tcell model is binary. You've got naive and TH2. Have you tried this method on a more complex developmental pathway? You have multiple waves of differentiation and concordant proliferation.
It's a very good question. So there is one data set about hematopoietic stem cells. Actually, with Betty Göttgens, they've been looking at more complex differentiation processes where that's the case. And there's upcoming data sets that looking at early embryogenesis, where there are multiple lineages that branching apart and there's more complex variation happening. So yes, that's on the go. I mean, this was the methodological development, but we also see good results on more complex processes.
Excellent work, brilliant. So I have a question about the use cases of this technology. So of course cells live in environments, right? So they live in communities. Mm. How important do you think is it to know what is going on cell by cell to infer biological functions and things like that? Because they are going to interact. And is it going to be the average of those expression levels and cell cycle states, or is it the individual cells and what they're doing?
It's a very good question. I think it depends on the process we're looking at. Certainly, I think the interesting insight is that we can do inferences about what happens at the level of individual cells, because very often we cannot measure that. We may be able to very, very coarse-grained measurements of the transcriptome, maybe a little bit of other omics layers. And hence, doing these inferences will be important, what happens at the single cell level. I think we have both events. We have decisions like differentiation that happen at the single cell level where lineages branch out. We need to know what happens for single cells. And there are other processes, for example, particular genotype-phenotype maps, where I think that bulk changes, a little bit of expression changes that happen on average in many cells are the more important phenomenon. So I don't think that's at least sort of my naive view at the moment, being at the beginning of this field, that we can take a single answer that will apply to all circumstances.
Hi, Oliver. I have a question about using your method in heterogeneous cell populations. So, for example, ES cells themselves are, either completely undifferentiated or partly differentiated, or you have these certain marker genes like CD4 in blood cells, and you might want to just treat one specific cell type or even GATA3. So, how do you apply the methods to tease out variance in cell population?
So it's a very good point. I mean, one of the really important things to take care of is, for example, if you have populations that do cycle and don't cycle, if you just naively apply this method without imposing that important biological knowledge, you may be explaining away variation that you actually be interested in. That's one of the main challenges. So you can fit this model, and I really briefly alluded to this with this T-cell differentiation process. You can fit this model using multiple gene sets. And there is some use cases in doing so, but also one needs to be a bit careful in knowing what one do. We describe it quite well in tutorials of what one needs to do. But the key point is, if you want to understand cell cycle, it is sometimes important to tell the method that actually there are two different cell types. If you just merge everything into one big bucket, you might get misleading answers. That cannot be avoided. But I think in many cases, you can, with very mild and obvious information that you have on the side, get very meaningful answers.
Hi, thank you. Very interesting talk. Do you think you can use this kind of approach to verify the annotations used in the first place to kind of subselect genes?
Whether we can look at better cell cycle genes? Yeah, that's a very good question. So we haven't attempted to do this. There's a separate thing we actually have done on the side now, which actually just came out 2 days ago, where we try to use similar approaches to not account for the cell cycle variance, but predict the cell cycle status of individual cells. And obviously one can go back using this predicted annotation and look at new marker genes. We haven't done this. You could do, my worry would be that it's correlative, and you might get genes that are hitchhiking the cell cycle variation, but they're not causative. So it's a starting point, but I think naive feature selection won't solve the problem.
Okay, our last question.
Hi, very nice work. Some data set dropout events and are kind of a problem. So this is essentially a normal model. Did you try to have some sort of inflation added to the model?
Very good question. Thank you for this. I mean, what I'm just trying to go quickly back to this. We are propagating technical noise variation through the model that we're estimating. So basically, we use a normal model that accounts for the technical noise variance that we do observe in the estimate here and an additional term in the model. But there are limits to this, and this will happen if the capture efficiency is not the limiting aspect, but also the sequencing at the end, when really in the Poissonian regime, then this doesn't work. So I think dropout we can handle. We cannot handle very sparse data sets as they're generating from drop-seq at the moment. And that's something we're actively working on, so there will be an extension soon that handles this really shallow sequencing data sets.